Ladies and gentlemen, we are here today to get down to business. See, I'm using my serious face right now. We are here to save your life today with
deoxyribonucleic acid.
Only one of the many types of nucleic acid. For those who are just tuning in, nucleic acids are the building blocks of living organisms, kind of like the backbone of society. Some other types include RNA, mRNA, and tRNA. Bill, can you tell all the good people at home what these nucleic acids do?
Well, Sydney, the basic understanding is that these cells help it replicate and build proteins.
Reminds me of money. Maybe this
deoxyribonucleic acid
will make you even richer. After all, you have to spend money to make money, right? Just like your bank account, these cells will continue to replicate and build even more and more proteins.
That's why I invested in good deoxyribonucleic acid, or DNA, a long time ago before I was even born. And since then, you can say I've definitely benefited from some good DNA. Thanks, Mom and Dad.
Now, if you're not fully invested and sold on this, then this will do it for sure for you. Everyone give a big warm welcome to Bill Nye, the smartest and greatest scientist to ever live.
Thank you for the wonderful introduction. You could say I'm so smart and great because of my good DNA.
Bill, can you please tell the wonderful people at home the basic structure of nucleic acids?
Sure thing. I brought a model along with me to help viewers understand. Two long polynucleotide chains composed of 4 types of nucleotide subunits: adenine, cytosine, guanine, and thymine. There are 2 types of base, purine and pyrimidine. For example, adenine bonds to thymine. Adenine is purine and thymine is pyridamine. The DNA structure is double-stranded and wound into a helix that is kept stable through hydrogen bonding. The double helix has 1 complete turn every 10 base pairs. Nucleotides are composed of 1 sugar, which is a 5-carbon monomer, bonded to a phosphate group and a nitrogenous base pair. Each nucleotide is bonded to another nucleotide on the other half, connected by a base pair. Adenine bonds to thymine and guanine bonds to cytosine. Each base pair is linked by hydrogen bonding.
Very interesting. Thank you, Bill.
Did you know that nucleic acid is responsible for hereditary information? There are so many variations that the DNA for each person is very different. DNA is what gives information about the living organism and is what causes each organism to be different from another. The sequence of the base pairs determines the information in the cells. The information from the DNA in the cell is passed to its daughter cells through reproduction, creating proteins known as genes. For the process of mitosis, DNA coils itself into chromosomes, which are what contains the genes. The DNA sequences are involved in determining when and how much of the proteins are made. The enzymes read the sequences and are then transcribed into messenger RNA, or mRNA. The ribosomes of the cells act as translators and read the messenger's codes, which help determine the order of amino acids to produce the proteins. It passes biological instructions to other cells and parts of the body. DNA is in the nucleus of the first cell created in an organism. For example, the chromosomes and genetic information is half of the parent and half of the other parent and become part of the first cell of an organism. In the nucleus of that cell is the DNA. It then copies itself during cell division. The replication of DNA is responsible for reproduction to maintenance and growth of cells, tissues, and body systems. When the cell is going through mitosis, the DNA in the nucleus divides itself down the middle, turning into the single-stranded RNA. When RNA is in the new cell, it replicates itself by using sugar and phosphate groups bonding to the correct base pair. A will bond to T and C will bond to G. The end result is a perfect match to the DNA before the division process. Sydney, could you tell some people at home why they have to have DNA?
Well, for starters, new genetic diseases are coming out every month, and DNA has a part to play in this. For example, Huntington's disease is the expansion of a 3-nucleotide portion of the gene, causing late-onset neurodegeneration and even death. Huntington's is affected by the dominant gene and affects the 4 chromosome, and in most cases of Down syndrome are related to trisomy 21, which means each cell in the body has 3 copies of the chromosome 21 instead of the usual 2 copies. This can be tested for with a non-invasive prenatal test, or NIPT for short, which is performed on maternal blood to screen pregnancies for the most common fetal chromosome anomalies such as trisomy 21. This test is performed on maternal blood samples which contain DNA from the fetus. And in 1994, the first gene associated with breast cancer, BRCA1, was identified on chromosome 17. And a year later, a second gene associated with breast cancer, BRCA2, was discovered on chromosome 13. Carrying a mutated form of either BRCA1 or BRCA2 means having an increased risk of developing breast or ovarian cancer. Children of parents with BRCA1 or BRCA2 mutation have a 50% chance of inheriting the gene mutation.
Wow, very interesting. I think everyone now knows the importance that good DNA has.
Certainly. Bill, can you please tell me and people at home where we can obtain DNA from?
To collect a sample of DNA from you or a friend, follow one of these 5 methods. Number 1, blood. Since the red blood cells have lost their nucleus, the DNA is found in the white blood cells. To collect the DNA, scientists must separate the white blood cells from the rest of the blood. They use detergent and an enzyme to release the DNA. DNA can be taken through the blood through whole blood, blood spots on paper, or dry blood.
I'm getting a message. And they told me that you can also get a DNA sample from saliva. In order to collect a saliva sample, you can't eat, smoke, chew gum, drink, or brush your teeth before collecting the sample. Rinse your mouth with warm water, then 10 minutes later, wipe the inside of your cheeks with a cotton swab. You can also do it collecting a hair sample. When collecting a hair sample to find DNA, you need to be sure that the bulb is still attached. The bulb contains some tissues, which is where the DNA is found. Without the bulb, the hair is useless for collecting DNA. Also, your bones. Obtaining DNA from the bones is often the most difficult way to retrieve it. Depending on the condition of the bone, it may or may not have retrievable DNA. It is usually taken from the shaft of the femur or humerus. And this I find the most interesting. You can actually get DNA from envelope and stamp. DNA can be found from the saliva left on the envelope after licking it to seal it, or stamp after licking it to stick it to the envelope. However, this method is sometimes the most unsuccessful due to the sticking agent on the envelope or stamp.
Some other ways to find DNA include sweaty t-shirts, used razors, earwax, fingernails, socks, urine, toothbrush, chewed gum, dental floss, cigarette butts, or hats.
Now that's a lot of ways to get DNA. Ladies and gentlemen, as you can see, DNA is the building blocks of all living organisms. Defective DNA can result in serious implications such as early onset neurological damage, muscular deformation, and even premature death. Please, I beg of you, if you are invested in your future, then invest in good DNA. Thank you, and good night.